Hello student. Now today I am going to explain a very typical topic of evolution, that is microevolution, which contains a very important topic, that is Hardy-Weinberg equilibrium. According to Hugo de Vries, evolution is of three-step formula, or you may classify the evolution into three groups. The first one is microevolution, which is at genetical level. It may either be visible or not. The accumulation of several microevolution will leads to macroevolution. The macroevolution is always reported at the formation of new species or species level. And the last one is mega evolution, which is above species level, that is class, order and phylum. But our syllabus is arranged with the first topic called origin of life. Then, according to available evidences, the second topic is evidences of evolution, which is directly related to mega evolution. The third part of the syllabus is theories of organic evolution, which is related to the origin of species, as you know about Lamarckism and Darwinism. And the last topic of our syllabus is microevolution, which contains Hardy-Weinberg equilibrium. Actually, in evolution, this is the first step. But on the basis of evidences available with the development of genetics and cytogenetics and the different tools and technique, it is the most sophisticated technique. It is the most sophisticated topic which is included at the last of the chapter. According to a very great scientist, Dobzhansky, it has been stated that nothing in biology makes sense except in the light of evolution. It means if you are going to study evolution properly, you should know almost all the basic concept of biology. And again, if you are going to study almost all basic concept of biology, you should know evolution. So the microevolution is directly concerned with the Hardy-Weinberg equilibrium. Hardy-Weinberg equilibrium. According to this Hardy-Weinberg equilibrium or law, if all the genetical condition remain constant in a large Mendelian panmictic population, the frequency of allele and genotype remain constant generation after generation. It means in any Mendelian, panmictic means random mating. It means in a large Mendelian panmictic population, if you consider the frequency of capital T is P, the frequency of small t is Q, then HardyWeinberg tried to state that P plus Q is always equal to 1. If we see the F2 generation in monohybrid cross, where the genotypic ratio is capital T, capital T, capital T, small t and small t, and put this frequency into the F2 generation, then it will be P square plus 2PQ plus Q square, that is P plus Q whole square. P plus Q is equal to 1, 1 whole square is equal to 1. This is simply stated by HardyWeinberg according to the law of genetics available at that time, that is Mendelian genetics. But if in all generation allele frequency and the frequency of genotype will remain constant, then what about evolution? Because evolution requires change in frequency. If there is not any frequency change will be there, the organism goes to stasis. No change in gene frequency will be static for the species form. It means the HardyWeinberg law is the supporter of fixity of species. But now in general life we see that the variation and evolution is a continuous phenomenon. The today test of HardyWeinberg genotype frequency are used primarily to test population stratification and other form of non-random mating. In that case, several genetical factors are there which can break the HardyWeinberg equilibrium, that is P plus Q is not equal to 1. These genetical factors which can prove the P plus Q is not equal to 1 will be the elemental force of evolution. And these factors are, the first one is natural selection, as stated in Darwinian natural selection, Darwinism. This natural selection is of 3 types, which can break the HardyWeinberg law, that is, it proves P plus Q is not equal to 1, and the first natural selection mechanism is number A. I will write the name later on. The example of the first one is selection of peppered moth in Europe against industrial melanism, and it was experimentally proved by Bernard Cattleville. The pipered moth is scientifically known as Biston betullaria. It is the white form, and its mutant form is Biston betullaria carbularia, which is black one. So if we put the graph against industrial pollution in Europe, you may say that whenever the Hardy-Weinberg law is applicable, three types of individuals are there in population. First one is capital B, capital B, capital B, small b, and third is small b, small b. And according to Hardy-Weinberg law, the graph will be like this: P square, 2PQ double, and Q square. But now before 500, before industrialization, about 500 years ago, when no industry is reported in Europe, all the bark and snow like white. And try to assume two types of moth are there over the rocks or over the ice. One is black, another is white. So the probability of white one is more for concealment. Black one is exposed, more and more killed by the birds. So selection of white one is there. In this case, the natural selector is bird. But when we plot the graph of this case, the peak of the graph is shifted towards Q square. But within this 500 years, after industrial melanism, due to the pollution, the bark and ice and other parts of the Europe are like this. It makes it smoky or dusty. Having two types of moth, one is white and another one is black. In this case, the white one is more picked by the birds and selection of black one is going on. So in second case, the graph peak is shifted towards the black one. It means before industrial pollution, the direction of selection towards the white one, and after industrial pollution, the direction of selection towards the black one. In formula, P square plus 2PQ is going towards 1, that is P plus Q is not equal to 1. In this case, Q square is going towards 1, that is P plus Q is not equal to 1. Because the selection takes place in a particular direction towards a particular phenotype, so this type of selection is known as directional selection. This type of selection is known as directional selection. Second, one in selection, the second one name will be given later on. The best example here is heterozygous selection for sickle cell anemia. In this case, there is selection of heterozygous individual, not homozygous of either side. As we know, as a simple graph of HardyWeinberg, let three types of individuals are there in a population: capital H, capital H, capital H, small h, and small h, small h. In these three types, this small h, that is Q square, having genetical anemia. So, its death is sure, almost sure. It means Q square is always decreasing. In this case of both dominant gene, the RBC is normal rounded. So no chance of genetical anemia here, that is sickle cell anemia. But these individuals are more susceptible to malaria. And as you know, in Asian and African country, thousands of deaths due to malaria is reported per year. So P square is also decreasing. But in heterozygous condition, where the RBC get slightly deformed, there is neither malaria nor anemia. So these individuals are selected in nature in better way and in large number. Further, the heterozygous breeding produces these two. But whenever there is heterozygous selection, the graph will be like this. The 2PQ is going towards 1. This type of selection is known as stabilizing or cumulative selection. In some book, it has been written that it is also known as balancing, but actually balancing does not have any relationship with this stabilizing selection. This stabilizing selection is simply heterozygous selection. But the point is, the Q square is decreasing and decreasing and decreasing because its death is sure. But the number of Q square is balanced in population due to heterozygous breeding. So the presence of Q square in population is known as balancing, and it is possible due to the heterozygous breeding. The third one is disruptive selection. In this case, third one is disruptive. In this case, selection of pure form of both extremes and elimination of heterozygous. If you again put the graph according to Hardy-Weinberg equilibrium, let the example of farming of goat, Copra hircus. Let 3 variety or breed of goat is available in farming, and according to Hardy-Weinberg law, the graph is like this. As you know, the total energy provided by nature at the time of zygote formation is limited. If the individual try to exhaust its energy in protein formation, a certain amount of protein can be formed throughout the life. Let apply here. If this type of goat variety, let it is Pashmina, it can produce better quality of wool. It is protein. If it produces better quality and large amount of wool, it can't produce better muscles or mutton. The second on in the plain, this type of goat farming is mainly for good quality of mutton, but it can't produce better wool. So the farmer available in the area of hilly area, they are trying to farming this pashmina for better product of wool and their products. But in the farmer available in the plain area, they are trying to farming this type of goat for better mutton. The heterozygous condition can neither produce better wool or mutton. So these type of breed are eliminated by both the farmers. Ultimately, in several generations and after pure farming, the graph like this, and now the graph like this. It means 2PQ is going to 0, P square is selected, Q square is selected, P square plus Q square is going towards 1, that is P plus Q is not equal to 1. In this way, the Hardy-Weinberg law Mendelian population get disrupted into 2 pure form, P square and Q square. So this type of selection is known as disruptive selection. This is the selection. The next important point is genetic drift, which can break the Hardy-Weinberg equilibrium. The first one is selection. The second one, 5 factors are there. The second factor is genetic drift. And as you know, drift means floating. Drift means floating. It can be defined as sudden change in gene frequency in a small isolated population. As population restricted to a particular area on a small island, or a small population where there is not any transfer of gene flow is not there, there is not any mixing of gene, Such type of small isolated population, whenever there is reported sudden change in gene frequency, the condition is known as genetic drift. The mechanism of genetic drift was explained by three different mechanism of evolution as reported by number one, Sewell and Wright. They were physiologists, but now, as you know, in biology, most of the events are the example of serendipity. So now Sewell Wright included in genetics or evolutionary genetics. In this case, they worked on African tribe who feed on dry beetroot having deficiency of G6PD, that is glucose 6 phosphate dehydrogenase. Actually, it is the enzyme of anaerobic respiration in RBC. In RBC, normally glucose converted into lactic acid under anaerobic condition. But if G6PD is absent, the lactic acid is converted into lactones. And when these lactones get filled with, associated with the lipid of plasma membrane, the RBC plasma membrane get fragile. This is the finding. Then what is the relationship of Sewell and Wright and this event? Let the example. If a small isolated tribal population is there in a particular area, according to HardyWeinberg law, all three types of genotype are available there. And all of sudden, there is attack of Plasmodium vivax. Its incubation period is 14 days. As you heard in case of corona, its incubation period is 1 week, 1 day to 1 week, or sometime 14 days. So incubation period is 14 days. So within 14 days, and mind it, it is tribal population. There is not any doctor or they have not any attitude to treat the disease. Just after 14 days, when malaria is spread in this area, all these individuals having normal G6PD gene are susceptible to malaria and going to death. But the Q square having deficiency of G6PD, whenever the Plasmodium try to enter into the RBC for erythrocytic cycle, the RBC get burst up. It means the individual having G6PD deficiency, the Plasmodium vivax is not able to perform its erythrocytic cycle. No formation of hemojoin is there, no malarial fever will there, no death is there. So this Q square individual, due to the deficiency of G6PD, having immunity against malaria, and their survival capacity is more and more. The second one is founder's effect. Second one is founder's effect. Let the example first. If a small isolated area is there and three types of individuals are available in that area: black dot, droplet like structure, and cross like structure. By any reason, the individuals get distributed or isolated in three different areas: A, B, and C. The first one is provided only with black dots. It means black dot is equal to 1. Sudden change in gene frequency. Here P square plus 2P cube plus Q square is equal to 1. Let black dot 1 is P square, then suddenly P square is equal to 1. Sum of the area is provided only with cross, then Q square is equal to 1. But sum of the area is provided with all these probable structures, so genetic drift is not applied here because there is not any sudden change in gene frequency. It means the founder effects is the loss of genetic variation that occurs when a new population is established by a very small number of individuals from a large population. It was first fully outlined by Ernst Mayr for speciation in 1942 using existing theoretical work by Hardy and Weinberg. But I am trying to explain another mechanism of founder effect. Let again a small isolated population is there. Here different genotype of individual is there. Other individuals are also there. By any natural calamities or mean, these individuals migrate from here to any new place. It may be several other places where the organism can't survive, died out. In evolution, dead organisms and the death of the organism is not valuable. Survival of the organism is valuable. So when these organisms try to migrate towards a new place does not mean empty place or bare land. Here all the species are there except X species. So when this new species migrated here, he may find suitable place for survival and shows fitness. In this evolutionary genetics, the term fitness means reproductive fitness. It means better survival of organism in new change area and production of fertile offsprings. Production of fertile offspring. So this X species shows fitness in this area, and now in some time it get flourished to the whole area. Then you may say foundation of a species in new area. Foundation of species in new area having a particular allelic frequency. This is called founder's effect. The third one is bottleneck effect. The third one is bottleneck effect. In a simpler term, I would like to explain you the bottleneck effect, but with graphs also. Bottleneck effect. Let any particular species is distributed throughout the area, and the best example according to our syllabus is Asiatic leopard. If you put the graph here, the number and